So evolution of protein synthesis, it involves both the evolution of the genetic code, the evolution of tRNAs, the evolution of tRNA synthetases, evolution of ribosomes, evolution of ribosomal proteins, and evolution of translation factors. So this is a very large and complex field. So translation is heavily dependent on the RNA molecules. We have the messenger RNA, of course, we have the tRNAs, and we have the ribosomal RNA, rRNA. So most of the functional aspects of protein synthesis are related to RNA molecules. And to go back to the history of the 60s and 70s, the ribosomal RNA was used to establish the relationship between species. So Carl Woese and George Fox published a paper in 1977 that said that there is not only prokaryotes and eukaryotes, there's also archaebacteria, because they comparing the ribosomal RNA, it was evident that they belong to an entirely different class of molecules. They were having very much the same type of features, but they grouped together in, and separate from bacteria and eukaryotes. So somehow, if we trace back, because as I said, the genetic code is the same and many of the molecular systems, so it all goes back to some organisms that preceded the three different things we know now. And it's called LUCA, not Lucas, but LUCA, last universal common ancestor. And before LUCA, there was maybe some prebiotic evolution, but you also know that there has been an old hypothesis asking whether our world, our planet, was infected from space by some spores, for instance. So this is still not ruled out. In such a case, there must be life on some other planet in our, say, our galaxy. Maybe there is no other planet that has harbored life. Well, maybe now that there has been observations that there is water flow or has been water flow on Mars. So maybe there has been life there, and now, of course, it will be very exciting to learn if that is so. But well, this panspermia hypothesis of infection from space remains a possibility. So in such a case, this prebiotic evolution would be less important. So evolution of the genetic code. The genetic code, as we talked about yesterday, is universal. But of course, there are some dialects spoken so there are some mitochondria in strange organisms which are different. But these are just minor modifications, minor dialects. So the genetic code is absolutely the same throughout. And the code is distinctly non-random. I will show you that to you soon. And the selective pressure to maintain the code is very high. Because, I mean, in a developed organism, if you would change the code, there are so many things that need to be changed, so this is impossible. So once the code developed, it had to be kept. And here's the genetic code. I don't think we need to show it again, but we just proceed. How did it evolve? Maybe there were fewer amino acids in the very beginning, or maybe the fidelity of protein synthesis was much less. And this we don't know. Thank you. And maybe there could have been alternative codes. If our evolution started from the prebiotic stage, then there could have been a number of different codes existing, and some of them were lost later on. maybe by some major catastrophe, or they were outcompeted. They didn't really support life in the way the current one has. So there are three main theories for the evolution of the genetic code. There could be a structural match between the anticodon and the amino acid. And so the anticodon here and the amino acid somehow would be able to chemically interact with each other and recognize each other. But due to the discussions that we have had over the last few days, you realize this is an impossible thought. They are so far apart that it is impossible. So another possibility is that the codon structure co-evolved with biosynthesis systems of amino acids. And this is a possibility. And another one is something called the error minimization theory, because certainly you would like to avoid making mistakes in translation, because mistakes, at least with highly evolved organisms, would be very dangerous. If you make rubbish proteins, then you wouldn't survive. And one statement which has been made with regard to the genetic code, maybe the code we have is a frozen accident. And actually, if you look at the code and look at the different amino acids, there is no physical explanation why a certain amino acid has a certain code. So it's very difficult to understand the connection here. But maybe this was just something that happened and outruled every other possibility. So it was an accident that was then captured. So this is the way we have to go. But this accident must have happened before Luca, since all domains of life have the same genetic code. So we have the tRNA, of course, it's the substrate of translation. You talked about that sufficiently. So how did the tRNAs evolve? Maybe the first type of activated amino acid was just an AMP with an amino acid on. And maybe this could participate somehow in making short peptides. Or we have the CCA ends of the tRNAs. The CCA is common to all tRNAs, and actually this CCA end is added by an enzyme to a ready tRNA. So the CCA end is kind of a modification added to all tRNAs. So maybe the CCA end could have been the early version of a tRNA, or maybe first AMP and then going to CCA. But of course, the CCA wouldn't have any opportunity to read different codons, because it would always be the same CCA. So, but at least it could be involved in making short peptides, short random peptides. But people have also discussed that maybe a fragment of the tRNA could have been an early version, say such as a mini helix of the acceptor end. So this part, the blue part here, could have been an early tRNA. We don't know. And evolution of the tRNA synthetases. As we discussed yesterday, there are two families of tRNA synthetases. And they must have evolved separately because they are entirely different. They could not have any strong relationship to each other. So, of course, this is the reaction that the tRNA synthetases go through in both families. So maybe the tRNA synthetases could have evolved from two ATP binding proteins composed of single domains. So maybe it was a single domain protein that was able to bind ATP and able to bind the amino acid So the whole thing started from the single domains because you need to start in a simple way. And what is common to both families here, they have one common domain, which is found in the whole family, the whole class of the tRNA synthetases. So there are 10 of the class 1 and 10 of the class 2, essentially. So there could have been just the single domain which was able to charge some type of short tRNA with the amino acid. This I told you already. And this I also said. Did the CCA trinucleotide precede the tRNAs as the functional adapter? Characteristic differences between the two classes we also discussed yesterday, so we don't need to go through them. The sequence motifs, the different subclasses here, just to remind you, the charged or polar residues are class B, aromatic in class C, and essentially most of the hydrophobic residues are in subclass A. And the amino acylation on the different hydroxyls here, the fold, and you remember the organization of the tRNA synthetases with one common domain, the green one here. This is the class 1 type, the Rossmann fold, and here's the antiparallel beta structure in the class 2. Now, we come to something one step forward here. And this is, again, the genetic code, but now we look at some further details of it. So the second position and the class of amino acyl tRNAs are related. So here, class 1 is painted in green. All these are class 1 and also the ones over here. And class 2 are the ones we see here. So there is, say, a non-random distribution of these. Particularly in the class 2, where everyone that has a second base C belong to the second class. And here are some further details. The aromatic residues, phenylalanine, tyrosine, and tryptophan, they all have a U as the first base. They belong to the top level here of the genetic code. So there are similarities. And the negatively charged residues and their derivatives, asparagine and glutamine, all have A as a second base. Glutamine, asparagine, lysine. So actually, there are two lysine tRNA synthetases. One in class 1 and one in class 2. I haven't mentioned that before, but this is the case. So there are actually 21. There can be 21 different tRNA synthetases because lysine has 2 different ones. And then aspartate and glutamate. So they all have an A as the second base. So this is lysine star belonging to class 2B. And furthermore, 4 out of 6 class 1A enzymes have U as the second base, and 4 out of 6 class 2A enzymes have C as the second base. And now this is an organization of the amino acids and the tRNA synthetases in this way. So you could say here down is the aromatic. So tyrosine and tryptophan, they belong to class 1C, but phenylalanine belongs to class 2C. So furthermore, the acidic ones and lysine, glutamate, aspartate, glutamine, asparagine, lysine 1 and lysine 2, they belong to 2B. And up here we have methionine, serine, valine, and threonine. they are very similar. So the methyl group, one of the methyl groups in valine is replaced by an OH group in threonine. Okay. So maybe they were all ancestors, and this is one hypothesis which I've seen in the literature. So there's a class 1 ancestor and a class 2 ancestor, just being the common domain. And if you make a relation here, oops. The two classes have related amino acid residues. That was what I showed you, the aromatic ones, for instance. So the amino acid tRNA synthetases from the two classes could bind to opposite sides of the tRNA, and I'll show you how that would happen. So maybe two molecules, two different domains would bind to one tRNA for efficient identification, protection, and catalysis. So here's the tRNA then, and class 1 one domain molecule would bind here, and the class 2 domain molecule would bind on the other side, because this is basically the way they operate. They bind to different sides of the tRNAs. And if we would make that experiment here, so here you have a tRNA, and if you bind then isoleucine tRNA synthetase, class 1 on one side and class 2 on the other side. And then you would bind glutamine tRNA synthetase on one side and aspartate tRNA synthetase on the other side. You could achieve this, and this is just rotated in a different way. Sorry, this is the same picture, and this is the same picture in two different directions. And here is also another one in two different directions. So these two domains here, they don't collide severely with each other. They could bind on their own side of the tRNA, and maybe this could be an interesting thought of how the tRNA synthetases were working in some much earlier state. But this is not proven. This is just an hypothetic observation. So evolution of the ribosomes. And ribosomes are present in all organisms. They must have an earlier history. I mean, how could you have the same type of ribosomes in all organisms unless there was an evolution, unless there was they had a common origin. And it's also very difficult to imagine sophisticated proteins before the ribosome had evolved. The ribosome is required to make the accurate definition of how the protein should be produced. But here we have a chicken or egg problem, because the ribosome requires proteins also to function. So how did it come about? I mean, if you need proteins to produce proteins, where do you begin? So again, you see the ribosome. You've seen it now a number of times. You don't need to spend too much time on that. So here is another picture you haven't seen yet. So this is from the side. So you see the small subunit here and the large subunit here. And there's the channel between the two subunits through which the messenger is threaded, and also the tRNAs are coming in from R side and then go in to the P side and then to the E side, and they are lost on the back side. And we said that the decoding site is here and the peptide transfer site is over here. And here's the peptide exit channel. So certainly the functional sites are shared between the two subunits. So the ribosome has several important functions. So one is the binding of the messenger RNA, and the active component here is the ribosomal RNA. We said that or we didn't say it, but it's true, that the Scheinen-Dalgarno sequence of the messenger RNA binds to the 3' end of the ribosomal RNA. So there is a nice base pairing possibility here that positions the messenger RNA in the proper place on the on the ribosome. But there's so much more to talk about that we don't have time for everything. Function binding of the tRNA, that is involving primarily the ribosomal RNA, but to some small extent proteins. The selection of the cognate tRNA, I mean the cognate is the correct tRNA compared to the incorrect one. And of course they all have to come in to test if does my anticodon fit with you? Okay, no, okay. And then I come back now. Oh yes, so then I bind. So selection of the correct tRNA. Here is also the RNA very much involved. I remind you of the adenines 1492 and 1493 and guanine 530. But there's a protein also, S12, which is maybe important. The peptide transfer, again, the ribosomal RNA and the tRNA, and maybe a protein, I'll come back to that, is participating. And the interacting part. So actually, let me see now. I think here you see the A site. Yes, no, this is the P site tRNA, the CCA end of the P site tRNA interacting with bases over here, and this is the A site CCA end of the A site tRNA interacting with bases here. But if you look now around this point, the CCA ends, they obey the two-fold symmetry, and the rest of this structure can be rotated such that this bit here folds onto that, and essentially everything of the red can be rotated and placed upon the blue. So the peptide transfer area of the ribosome has a two-fold symmetry, but it is only one sequence. So how can this happen? How can you have a two-fold symmetry? How can you evolve a two-fold symmetry in a chain of nucleotides? Well, there is one way, and this has been observed in proteins many times. If you take, for instance, the HIV protease, this is, of course, a very important protein which is being worked on to be inhibited. This is a dimeric structure. So it has an active site across the dimer surface. But there are enzymes which are strongly related to HIV protease in other organisms. And here the peptide is just one single peptide, but they remain dimers because there are two domains which are identical and they are related by a two-fold symmetry. And there are also cases where there are more domains which can be related by some symmetry. So I think these have the origin in monomeric, the monomers which dimerize, monomers that dimerize in this way, but then somehow in evolution it turns out to be better to have gene duplication and to modify the two parts of the active sites separately from each other, so you can get a more efficient enzyme in this type of fused longer protein. So the two domains, the one sitting here has some components here which fit better than if they would be the same, and the one sitting here has some different residues to participate in the active site. So if you understand what I mean here, maybe the early ribosome would have just a simple structure like the blue part. Maybe this part of domain 5 could have been the earliest ribosome, but it would dimerize. So there would be one monomer here and one monomer here. and they would bind to each other in this way, and they would operate with some very simplified tRNA-like structure to produce maybe dipeptides or short peptides. So this could be the structure of the earliest ribosome. Maybe this has not been proven by any means, but it remains a possibility. And I mean, how do you evolve a twofold symmetry other than the way I have described? I don't know how that will be possible. So I think this is one of my favorite ideas, that this could be the earliest ribosome. Maybe no more than 90, 100, 110 nucleotides per monomeric site. So this is what I've said already. Sideways movement of the tRNA, 180 rotation of the CCA end. The peptide transfer activity needs it. The conserved CCA ends of the tRNA are single-stranded and can rotate. The peptide transfer site is rigged up to promote it. But how is this achieved? And this is what I already have told you also. The symmetry of the peptide transfer center goes right through the central loop of domain 5. So actually, the A site part is sitting on this side in the central loop of domain 5, and this is the P site part. But if this has been the case, it would have looked simpler from the early states. And this is what I already told you. How do you evolve an 180-degree rotation? And this is seen repeatedly in proteins. Early peptidal transfer could have had two identical subunits related by twofold axis. A gene duplication, gene fusion would retain the 180-degree rotation and allow a differentiation of the two units to peptide transfer. Now, let's look at the ribosomal proteins. We have ignored them so far. And here I have, with E, marked eukaryotes. With A, I've marked the archaebacteria, and B, I marked the bacteria. Here, bacteria has 56 proteins, archaebacteria has 68, and eukaryotes have roughly 80. So this illustration shows how many proteins which are common between them. And bacteria, archaebacteria, and eukaryotes have essentially 33 proteins in common. So the ribosomes is not only the same basic structure of the RNA, but the same basic arrangement of the proteins. 33 proteins are the same between them. They're sitting in the same place, and they have sequence similarities, similar structures. But then bacteria has 22 proteins of its own. Archaebacteria has 11 proteins of its own. There are 35 proteins which are common between archaebacteria and eukaryotes, but bacteria and eukaryotes have only one protein in common that archaebacteria doesn't seem to have. And bacteria and archaebacteria have no further common protein together. So anyway, there are a good number of very much similar proteins between all species. Now, these ribosomal proteins, they look very strange. So this shows the structure of some of them. And what we see here, well, the globular domains, to the extent they are exist, they are shown in green, and then they have long extensions. We talked about the histones. Histones have very long N-termini, which are flexible, but also ribosomal proteins have extended structures. So these loops and tails, where are they placed in relation to each other? And maybe you could almost guess what I'm going to tell you. The tails and loops of ribosomal proteins are always conserved in the interior. And the folded domains are always sitting on the surface. And I didn't make it very clear, but if you compare the ribosomal RNAs to each other, it turns out that the parts which are lost, if you go to mitochondria, they are sitting on the surface. And the parts which are added in eukaryotic ribosomal RNA is always added, of course, on the surface, because in the interior there is no more space. So the changes are on the surface, and likewise the globular domains are sitting on the surface, but the tails are always in the inside. So maybe you could make the conclusion that the tails are the conserved part, and they're sitting in the inside in the very ancient type of early ribosomes. So 31 of the ribosome, 54 ribosomal proteins in bacteria have tails and loops. 23 of the universally conserved proteins have tails, and 14 of the 23 non-conserved also have tails. So are these tails and loops then the earliest proteins? So they, as I've already said, may be remnants of the earliest peptides. Now, onto a different topic here. And there's been a discussion of whether the ribosome is a ribozyme. Is the ribosome a ribozyme? Well, it turns out there is one protein which is called bacterial protein L27. And it is sitting here in blue, BL27. It has a long amino terminal tail going in, and this is now the green A side tRNA and brown P side tRNA. They're coming next to each other, and here's then the peptide transfer occurring. And this N terminus is coming very close to the loop, or close to the site here. So actually, if you remove some amino acid residues by mutation of this, then you see that the activity in the peptide transfer site is decreased. Not completely, but it is decreased. But it doesn't really reach up all the way to the peptide transfer site, so it's in the vicinity. And maybe it is stabilizing the CCA ends here and the ends of the tRNA, but it doesn't really engage in catalysis. So there's a stabilizing factor. There's another protein here also, universal L16, which reaches up in the direction of the peptide transfer center, but not all the way. But now look at this B here. B says this is only found in bacteria. So it's not found in eukaryotes or archaebacteria. So if this is an important protein, it should have been conserved between all of them. But this is not the case. So I think to say that this is, making the ribosome not to be a ribozyme is to stretch yourself too far. So it looks very much as if the ribosome can most comfortably be called a ribozyme. So in the discussion of the RNA world, of course, this RNA world must have preceded the DNA, RNA, protein world, because DNA nucleotides are made from RNA nucleotides by ribonucleotide reductase. And furthermore, ribozymes are very efficient catalysts. So the early ribosomes could hardly have been composed of RNA alone, because it would be very inefficient to make new proteins. So maybe peptides or small tail-like proteins would increase the efficiency of the ribosome. It would stabilize both the ribosomal RNA and stabilize the binding and the ligands here, the early tRNAs and the amino acid, so they would have a stabilizing function. But such stabilization would probably increase the rate quite significantly. So, as has always been said, evolution must have started from a simple system that evolved into something more complex. At an early stage, the biological world might have been based on RNA as the primary polymer. But could we call this the RNA world? Maybe not. I think such a world must have existed before the evolution of cells. And protein synthesis was a central event in the evolution of life, because proteins are always very important. So if one should say that the ribosomal RNA is participating in the catalysis is not entirely obvious. The hypothesis of the RNA world would require an RNA genome, RNA ribosome, a tRNA with two codes, the amino acid charging, the charging by the tRNA synthetases, and the codon-anticodon interaction. Could there be a ribosome amino acid tRNA synthesis? Yes. In vitro evolution has shown that this could be possible, that you could have an RNA molecule to catalyze the addition of amino acids to tRNAs. And you will also need an RNA replicase. And in vitro evolution has also shown that this could be possible. But to have a ribosome-based metabolism would seem to be going a bit far. And small peptides, as I keep saying here, would make a big difference because they would stabilize and increase the rate of all such processes. So in conclusion, this is the last slide. The RNA must have been a very essential ingredient in early evolution before LUCA. However, it had a strong need of a complement of peptides or small proteins. So I think this was the very end. And we could call this world, the early world, for an RNA peptide world. This is my point. And now I'm ready to answer any questions you may have, if there are any. You're buying the whole package? So this is very far from the fields I've been working on. And microRNA, yes. And I think I know very little about microRNA, and I suppose most what can be known remains unknown to everybody. But I think if your question is how can microRNAs be markers of some specific, I think a cancer cell is behaving in a way that normal cells does not. And maybe the microRNAs here have a very important role in say, making the eternal life of cancer cells. I don't know, but somehow there is some relationship. But I don't know, and I don't know if anybody knows.
Thank you. Thank you. Thank you very much. Because this is his last day, so I guess we can finish the class earlier. And for the past 3 days, I would like to appreciate your time and your energy, to share and to lecture us with your very nice lecture. So thank you very much for your coming, and please give applause to him. And as a thank you, and thank you very much. And of course, thank you for everyone for coming to this lecture series. I hope it is beneficent, and will be good for your. Maybe you will do next research on this field maybe. Yeah, thank you very much.
So actually, we haven't said this before, but I could possibly provide you with this material in a PDF format, so you can read it and think about it.
To be convenient, we will upload it on the website, so everyone can just download it. Is it fine?
There is a slight problem to put it on the website because I've stolen pictures from so many different scientists. So a paper distribution for those who want, or just on, or I mean by emails, or by yeah.
Oh, okay.
So because I put it on the website here, I would become a criminal right away. Maybe I am a criminal already.
Okay, so if you need some materials, you can just contact the secretariat. I think you know the email address, right? You can ask them, because I don't think that we have your email address. Kalian mencatat email address-nya? Ada. Oh, okay, we will send to your email. Okay. Thank you very much for coming.